All right, students, for our unit assessment, you guys are going to be reading this article that describes an experiment performed by Dr. Aaron Wheeler. Based on the article and based on his experiment, you're going to be filling out this graphic organizer. Okay, I would like for you guys to use the model graphic organizer that we used in class. Okay, so you're going to be assessed on these 5 different parts. The first part you're going to have to do is identify the research question. This is the question that you are trying to answer. So in this case, it's going to be the question that Dr. Wheeler is trying to answer in his experiment. You are going to sketch and label the lab setup. Okay, for this, you don't need to include any pictures, but you're going to describe how you would test this, what materials you would need, and what the setup would look like. Okay, so explain to me what Dr. Wheeler did. How did he test it? What materials did he need? And what did his setup look like? You're going to tell me over here, record the data, what measurements you would take if you were doing this question. Okay, you're then going to identify your variables. Okay, your independent variable. Okay, this is what you are testing. It's the thing that I change if I'm doing the experiment. Okay, so we could think of it as the thing that Dr. Wheeler is changing, or we could think of it as the cause. Okay, your dependent variable is the results that you're looking for. It depends on the independent variable. It is the effect. Okay, the last requirement for you guys is to list 10 constants. Okay, these are things that need to be kept the same. Okay, so reading about Dr. Wheeler's experiment, and you are going to identify all of these parts of his experiment in your graphic organizer. Okay, number 1, identify the research question. 2, sketch and label the lab setup and record the data. 3, identify the independent variable. 4, identify the dependent variable. And 5, listing your constants. Okay, so let's go to the article now, and I'm going to read this to you. Okay, up top, please type in your name, type in today's date. Part 1, directions. Read the article below that describes research being conducted by Dr. Aaron Wheeler. While reading, please highlight and annotate the article. Okay, you will not be graded on your annotations, but it will be helpful if you do this for the part where you have to fill in the graphic organizer. Okay, the title is Amazing Worms. Dr. Aaron Wheeler loaded a small wire loop with bacteria, lowered it into a dish, and waited. In moments, a tiny worm climbed into the loop to eat the bacteria. Dr. Wheeler placed the worm into a maze and started his stopwatch. The worm was tiny, only about the size of a dash. But Dr. Wheeler was using the worm to find out more about how people learn. Why worms? When scientists want to learn something about humans, they often study smaller animals first. In this case, Dr. Wheeler's worms are very simple. Scientists know exactly how many cells are in each worm and what each cell does. They know what chemicals are in the worm's brain. The information makes it easier to figure out cause and effect. If scientists can figure out which chemicals affect learning in worms, they can see if the chemicals work the same way in humans. The worms. The scientific name for Dr. Wheeler's worms is Cannelura habitus elegans, but scientists call them C. elegans for short. In nature, C. elegans live in gardens and compost bins. They hunt through the dirt for bacteria to eat and hitch a ride on a millipede or a slug when they want to get to new hunting grounds. They can't hitch a ride in Dr. Wheeler's lab, though. They have to find their way through the maze all by themselves. Pint-sized puzzles. First, Dr. Wheeler had to build a maze for his tiny worms. He used a machine that normally builds computer microchips to create thin, rubbery mazes about the size of a postage stamp. Each maze was shaped like a T, So the worms only had to make one decision: turn right or turn left. But the worms don't have eyes, so even the simple maze was hard for them to figure out. Dr. Wheeler always placed the worm's food, a clump of bacteria, on the right side of the maze. Each worm had 5 chances to find the food. The first time, the worms averaged about 15 minutes to get to the food, said Dr. Wheeler, but they finished in about 5 minutes by the 5th try. Even when Dr. Wheeler stopped putting food in the maze, the worms kept going back to the same spot. They had learned. Human learning. People can learn in much the same way. The first time you read a difficult word, it might take you several tries to figure out what it says. But each time you see the word again, you will figure it out more quickly. Soon you will have learned to read the word instantly. Some people have a much harder time learning to read. Math, memorizing lists, or figuring out puzzles may come easy for them. But even when they read the same word over and over again, they may have a hard time remembering what it says. This is called a learning disability. No one knows for sure what causes learning disabilities, but scientists suspect that certain chemicals in the brain may be involved. One major brain chemical is called dopamine. There's a hypothesis, said Dr. Wheeler, that dopamine helps with learning. A chemical connection. Did dopamine help Dr. Wheeler's worms learn? He decided to find out. Other scientists had a group of worms that were just like the ones Dr. Wheeler was using, except for one thing. The new worms didn't make dopamine. Dr. Wheeler repeated his experiments. He used normal C. elegans as his control group. He measured how fast they found the food and how long they remembered where to go after the food was gone. He then tested the worms without dopamine. Those worms were able to find the food fairly well. But when the food was removed, the worms without dopamine forgot their knowledge much more quickly than the normal worms. The research supports the hypothesis that dopamine helps with learning, said Dr. Wheeler. In the future, we may be able to use that knowledge and, he gestured to the worms in the maze, these little guys, to help people with learning disabilities. Okay, part 2, directions. Fill out the planning and carrying out investigations graphic organizer based on Dr. Wheeler's experiment. Okay, so my students, based on what you read, you are filling out this graphic organizer. Okay, again, identify Dr. Wheeler's research question, label the lab setup. Okay, tell me what his lab setup looked like. Tell me what his data looked like. Identify Dr. Wheeler's independent variable. Identify his dependent variable, and try to list as many constants as you can. If at any point you are stuck or you forget what one of these parts of our investigation are, feel free to go back to the model. Good luck.